Alright guys, welcome to Subu Medicine, and today we'll be discussing about the glycolysis. And glycolysis is one of the most ancient and also the most understood metabolism pathway. Alright, so that's what we'll be focusing on today in this lecture. So, you guys often hear this term glycolysis, right? But there's also another name that is given for the glycolysis, and that is called, let me just write it here, it's called Embden-Meyerhof pathway. All right, and it's based on the scientist. All right, and this is just extra. I'm giving it to you because you always get extra here, right? So look at the term glycolysis is. What is glycolysis? So basically, look, glyco. If you break down this term, the glyco is referring to a sugar, and lysis is breaking down. So you're breaking down sugar in order to what? In order to generate energy, and you generate energy in the form of ATP. That is what glycolysis means, all right? So, look, you're breaking the glucose, right? So you have to know what a glucose looks like. And glucose are usually in your C6, right? H12 and O6, all right? You have 6 carbon molecules and you have the hydrogens and O oxygens. So if you look at this, right? If I could make this, structurally, if I want to make it, you can just look at this one. 1, 2, 3, 4, 5, and 6, right? And then basically you're going to have CHO here, and you're going to have CH2OH right here, right? And basically, look, now here, after that, what is going to happen? You can make this up, okay? Bond like this, and then basically, look, you're going to have OH right here, H right here, right? And then I'm going to skip the third part right here, but let's just remember this, OH right here, and that's going to be H and H. Look, and then look, there's a 1, 2, 3, 4, 5, 6 here, right? And then in a carbon number, this is if you label this, is going to be 1 here, 2 here, 3 here, 4 here, 5 here, 6 molecule. And the carbon number 3, what is going to happen? This H is going to be here, right down here, and the OH is going to be down here, right? And look, this compound is what's called your glucose, right? So this is a glucose, but what kind of glucose? That is something you have to remember. Remember, there's also if you look at this middle of the glucose right here, all right? If your hydroxyl group is on the right side of this carbon, then we call them a D-glucose. All right. Now, it's important to know this too. All right. Let me make another one. Let's write down here CHO here. 1, 2, 3, 1, 2, 3, 4, 5, and then 6 here. Right. H2OH right here. Right. And then same thing. Look. Basically, you're gonna have OH here, and then here H here. Right. And then same thing with here. I'm gonna just make it have here H, carbon number 3. And OH here, all right? And then here, sorry, this is H, so this is OH, this is, okay, now here, let's make this as OH right here, all right? And then H right here. But now, look at the carbon, look at this carbon right here. On the carbon number 5, I'm gonna flip this hydroxyl group right here on this. So I'm gonna put it, hydroxyl group in right here, and then I'm gonna put what? H right here, right? Now, again, what is happening here? Again, this is what? It is a glucose, right? It is a glucose compound. But what is happening here is that, this is if I put here on the 5th position right here, this is the center of my carbon, and my hydroxyl group is on the left side, all right? Because on the left side, we call them L-glucose, all right? Now, what is the point of this? The importance of this is this compound, all right? These guys are enantiomers of one another, meaning that there's a mirror image. And this L and D glucose, these are based on the hydroxyl group located, all right? If your carbon is on the center, and on the right side of the center, right side of this carbon, if this hydroxyl group is attached into it, we call them a D-glucose. And that is what usually found in nature. That's what we try to, that's how, this is the glucose that we run glycolysis pathway. All right, and this D and L, this is, I'm not going to go too much on the organic chemistry here, but this is, this is a configurations, all right, that is based or defined by something called Fischer projections, all right? Now, this is an enantiomer of one another, and this is the D-glucose is the one that, you will run glycolysis, because this is what you found in nature, all right? Now, that is something that I had to quickly mention about it. But now, After this, let me put the structures, and also, let's say in a cyclic structure, how does this look like? And just for you to know this, right? It's important to know this too. So if I make this like, let's say glucose right here, all right? Here, and then look, there's a 6 carbon. If I label 1, 2, 3, 4, 5, and then you have a CH2OH here right here. Look down here, all right? I could make OH group right here, and then this guy right here, look, what is gonna happen? I'm gonna have OH right here, and then H here. And the carbon number D, this is the third, OH right here, and then H right here. And then obviously, right here, you're gonna have OH, and then H, and there's gonna be H. Look, this is how, in a ring structure, this is how the glucose structure looks like, all right? So this is a compound. And if I switch this, all right, if I switch in carbon number, like let's say in the 5, in this carbon number 5, if I switch it, all right, just like this, then we can make a L-glucose too. But this is a D-glucose, all right? This is how it's structurally, you can look at it in the cyclic ring structures also. Now, like how do we get glucose, right? That's what I have to talk about. Now, glucose, here's the word. You can found, all right? You can found glucose from where? Obviously, it's a monosaccharide sugar, and they are what? Aldehyde sugar, right? Because glucose monosaccharide sugar is an aldehyde sugar. So basically, let's say glucose, or you have a fructose, which is a what? Which is a ketose sugar. What does that mean? Which basically means that if you look at the structure, this just have a double bond with a ketone group into it like that, all right? And the aldehyde would be just like double bond with oxygen with the hydrogen attached into one of the bond, all right? This is aldehyde, and this is a ketose sugar, and also you have to remember galactose too, all right? And that is also aldehyde sugar. So all of them, like your galactose, what is going to happen? When you take that, you can take in galactose diet, fructose, right? And then when you take it, right, this galactose and fructose, they can, in the liver, they have enzymes to break this down into what? All of them down into glucose. All right, this is. So this is how, we take the glucose in our body, right? Now, we have to talk about, what is the importance of this glucose, right? Why do we take it? Glucose. Because these glucose are coming from what? Carbohydrate sources, right? And most importantly, we take this glucose, this carbs, is what? To generate energy. That's how we're taking. And these glucose are usually used for, right? Like functions of glucose are usually used for, let's write down a couple of things, okay? One thing is that, you run the glycolysis to generate ATP, all right? That's one thing you do. Other thing you do is, glucose are very, very important for like making a biosynthesis of, let's write down here, biosynthesis of like those chain of polymers. Like for example, your extracellular matrix, you find those molecules like polysaccharides. Okay, so for in order to make this extracellular matrix, this glucose are used there. Not only that, like glucose are used for what? Glucose can be stored, all right, in the liver or in adipose tissue as a glycogen too. So later, if the glucose level is falling down, we can use the stored glucose for the energy. Okay, that's what it does. Glucose, there's also a lot of glucose is there. We can actually use this glucose to generate this pathway called ribose. We can make ribose 5-phosphate, and from the ribose 5-phosphate, we can run the hexose monophosphate pathway shunt, and we can make a lot of this something called NADPH, guys. It's pH, okay? So NADPH, you can make that. And we'll talk about the importance of this NADPH in some of the lectures. But if I want to quickly mention something, this NADPH are very, very important for for fatty acid synthesis. All right, you should remember that. So these are like some of the roles of the glucose. All right, now after this, what I have to talk about is that, look, let's talk about this glucose. So your glucose, when you take it right here, right? Let's say you take it from the diet. Let's say here, you take a glucose on a diet, right? From here, the glucose is going to get what? Reabsorbed by this liver right here. That's where it's going to go. The portal vein systems and all that stuff, right? And then this is going to come out glucose, and then let's say the cells is going to take this glucose right here. Usually here, the glucose can in the liver, the glucose can store as a glycogen there, right? Now, other thing you have to remember is that whenever this is the pathway we're running, right? Whenever we are talking about the glycolysis, let's see what is happening here is that, look, this is a lysis. You are, okay, breaking down this glucose molecules in order to generate ATP, right? Not only this, in the process of this, you also generate, okay, because you basically you're oxidizing these glucose molecules and you generate ATP, and also you also make like NADH also, right? So this process of when you're breaking down and then you make ATP and NADH, this process, right? This process in biochemistry, we call them a catabolism, right? And then let's say after these catabolisms, right? And obviously you release carbon dioxide there, right? Then after that, what is going to happen is that this energy, right? This energy substrate, they're used for biosynthesis to do certain things, okay? And that is what is called anabolism. All right? So basically you're breaking down and you're building something, right? Like for example, you run the glycolysis to make ATP and NADH, right? You use this ATP and NADH to go to the Krebs cycles, and then also it goes to electron transport chain to make more ATP, right? So that what is that called? That process is called anabolism, right? So this difference between catabolism and anabolism, that you should remember that. Remember when you are breaking down glucose, you're gonna release exergonic energy, all right? That's what you're gonna do. Now, whenever we talk about the glycolysis, glycolysis is a two-path, all right? So one we can think about, one is investment phase and other one is a generation phase. Like in a simpler term, like for instance, right now you're watching me and learning the lecture of the glycolysis right now, right? So you're putting your time and effort into it, right? This is your investment phase. But then later, when you have generated the knowledge, right, you're going to implement that knowledge somewhere else. That is your generation phase, right? So you can look at that glycolysis, you can look in that way, right? So now, let's take this glucose molecule, which is a 6 carbon molecules right here, right? So basically you're making like, look, six carbon molecules, right? And then obviously you're gonna have what? You're also gonna have like NADA plus, right? You're gonna make 2 NAD plus, and then also like 2 ADP. And later you're gonna make what? You're gonna break down and you're gonna make 2 pyruvate actually. You're gonna make 2 moles of pyruvate, and we'll talk about how this will happen. Not only that, you also make the 2 NADH, and then you're gonna make 2 ATP. Also, you're gonna make 2 water, guys. You're also gonna make 2 water. Okay, and you also make a phosphorus. So basically, the most important you have to remember is that, all right, with this carbon, with the glucose molecules, look, with the 6 carbon molecules, we break down 2 moles of pyruvate, and pyruvate is a 3 carbon molecule, all right? So now we have to use this, and let's go in detail and how this works, all right? Now, so let's say, like for example, like you took the carbohydrates, the glucose, right? This glucose, what is going to happen is that you take the glucose, and this is like one of the cells, right? And also one of the things we have to know is that, why does the glycolysis run? Like, you have to remember this. Glycolysis takes place in cytoplasms of the every cells out there, all right? That is something that you have to remember. So this is, just think about, this is one of the cells right here, all right? This is one of the cells of our body, and this is where we're going to run the glycolysis right here. So when it comes right here, because the glucose is let's say it is a water soluble, all right? So it cannot just diffuse through the different cells. It requires special transporters, and there are types of transporter located in different places. So those transporters usually referred as, all right, usually they're referred as like a GLUT. These are protein transporter. Let me just write down here. All right, they refer as a GLUT, Z-L-U-T, GLUT 1, GLUT 2, and there are like about 14 of them. 14 of them that we know, but we're not going to go all of them. We're just going to go few that are important to us. And there's a GLUT 3, and then also GLUT 4, and then GLUT 5. All right. So remember, this GLUT 1 transporter, all right, these are insulin independent, and these guys are usually found in, all right, these are usually found in where? Okay, one of the things you have to remember is that, all right, we'll just make it in a small diagram here. So basically, if I quickly draw, what is this? They're finding basically a blood-brain barrier. You can think of it that way. All right. They're also found, this GLUT1 transporter, they're found in, what is this organ right here? Red blood cells. All right. GLUT1 transporter. Not only there, these guys are also finding like this. What is this? What am I making right here? This is a colon, guys. They're also found colon. And they're also found in placenta, right? So that's where the GLUT1s are usually found. This GLUT2, all right, let me write down here. Okay, it's found in the pancreas. Let me just write down pancreas here. Especially in the beta cells of the pancreas, that's where you're found. You're finding kidney, all right. So you find a kidney, this is my kidney right here. They're also finding your small intestine. Let me just write down SI with the small intestines. And guys, you don't want to forget, what is this organ right here? Liver. Liver also has a GLUT2 transporter, and you have to remember that, right? And then what about this GLUT3? Okay, here, I'm just going to write in placenta. That's where you find there, all right? And where else? Placenta, obviously you can find in neurons in the brains, right? You're going to find the small intestines. You're going to find in kidney as well. And then GLUT4 is very important, and GLUT4 is insulin-dependent transporter, meaning that in the presence of insulin, this GLUT4 transporter will come to down here on the cell membrane, so that way it can take more glucose in, right? And these are usually located in like, heart, mainly, you should remember adipose tissues, all right? Adipose tissues, and then heart, adipose tissue, and skeletal muscles. You should remember this, all right? So GLUT4s are insulin dependent, which are found in, all right, heart, adipose tissue, skeletal muscles. MUS is skeletal muscles, and the GLUT5s are for the fructose transfer, all right? Transfer. All right, so let me quickly repeat this, all right? Look, these GLUT1s, which are found, the GLUT1 found red blood cells, colon. They also find the blood-brain barrier. They're also found in placenta, all right? And this GLUT2 transporter, which is the beta cells of the pancreas, which are located in the islet of Langerhans, all right? That's what they're found. They're also found in kidneys, all right? And then also in a small intestine they're found. These GLUT2 transporters are. And don't forget, your livers are also, this GLUT2 transporter located there, all right? And then GLUT3 transporters are also located there, placenta, small intestine, neurons, kidneys, that's where these GLUT3s are located. And remember, this GLUT1 and GLUT3, they have a very, very high affinity, all right, for glucose. And GLUT4s are insulin dependent, which basically means is that this GLUT4 transporter are located in heart, adipose tissue, and skeletal muscles. And they only, these guys are very, very dependent on insulin, all right? So that is something that you should remember. And the GLUT5 is, as I said, is for the fructose transfer, all right? So when this GLUT2, all right, because these GLUT2s are always located in the cell membranes of the cells, of these cells. So if we talk about this one right here, right, the GLUT transporter, this cell takes it, right, and then usually GLUT2 transporters are, especially in the liver, they're bidirectional, meaning that when the cells comes in, what is it? They cannot go back. Like they can go back, that's what they do. So here, the glucose come here, this compound is one. Let's say, let me make this as a, all right, let me make this as a glucose right here, all right? So you have we can simply say, this is, let's say, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. This is a glucose compound, right? So when it came here, it can actually, because it is a water soluble, it can actually, diffuse throughout. So if this diffuses out, all right, through this transfer, let's say, with this transfer, it can go this directions, then we would not able to run the glycolysis, right? Because of its nature, because it's a water soluble molecule. So when the glucose comes to the cells, what do we want to do? We want to trap it. And how do we trap it? We trap it by putting like a charged molecule. That's what we have to do. We have to do with this. So that way the glucose can get trapped, and then we can use that glucose to break down to generate more ATP. That's what we do, right? So let's say this cell takes it, right? So when this cell takes it, what is going to happen? When this cell takes it, this is 6 carbon, right? Now let's say this is a glucose. I'm just drawing this as the same, all right? This is the same. All right? What is going to happen here? There's a special enzyme that's located there, all right? And these enzymes are very, very important enzymes. So let me write it down here. 2 enzymes I'm going to write down here, okay? And then, okay, let me make this as, all right? So you can think about this way. So what is this enzyme right here, all right? And then, okay, I'll just make this as I right here. All right. So there's one enzyme right here, all right? So the 2 enzymes are located there. So this enzyme we can simply refer as we can simply say, okay, this is what? we can say this is a hexokinase. All right, now, okay, let me erase this part right here. There's also another enzyme that's located there, okay, and that is called, let me draw it in different color. We'll just say, now this is another enzyme. This enzyme is called, let's say, glucokinase. All right, guys, it's very important that you know there are two different enzymes, right? Hexokinase are present in almost all the cells, right? But glucokinase are not present everywhere. They're specifically located in certain places. And hexokinase are present in cytoplasm. But glucokinase usually they're bound with this glucokinase, okay, binding, glucokinase regulatory binding protein, and they're usually in the nucleus of the cells. And whenever there's a high level of glucose is there, whenever the concentrations of glucose is going up, that's the only time the glucokinase will come. And these are only located in the certain part of the body, and we have to know where they are. The glucokinase is really important. You know this. Glucokinase are usually located right here. I'm going to write down here, okay? Liver, you should remember always liver, right? That's where they're located. And then obviously the beta cells of the pancreas, that's located there. And the other thing that you should remember is also located in the hypothalamus. It's located there, right? So these are some of the places, like, the glucokinase are located. Liver is very important. Beta cell of the pancreas and hypothalamus. You want to remember that as well, all right? And hexokinase is located in, other parts of the body. This glucokinase is also called hexokinase 4, all right? So now, anyways, so when the glucose is coming down here, right? These are water soluble, so this specifically enzyme is there, and what they do is they do a kinase activity, meaning that they'll put a phosphate group into it. And when you put a phosphate group into it, because phosphate is a charged molecule, these guys are no longer able to go out of the cells, so they get trapped right here, right? So this hexokinase, what they do is that they utilize, we have to know this, they use ATP, right? Because remember, and then they convert ADP, right? Look at this. What they did is that, now this is an investment phase. What they did is they take one phosphate group here, okay? And then they just put this one, because look, this is what ATP stands for, adenosine triphosphate, right? So when they, this hexokinase, what it did is it took one phosphate group and put it in here, this one right here. And this is gonna be right here, we'll make this first. And that guy right here is a phosphate right here. They put this PO4 3-. That's what they did. At a carbon number 6, that's what they did. All right? And one more thing you have to remember is that they use ATP, right? And they convert to ADP. The lower energy molecule is ADP is a lower energy molecule. But remember one more thing. I have to mention something about magnesium also. Let me quickly talk about it. Sometimes they don't mention the role of magnesiums. What is the role of magnesiums is that? Because what are enzymes? Enzymes are very, very important for what? For lowering the activation energy, increasing the rate of reaction, right? So this magnesium plays a cofactor, okay? And it plays a cofactor. It sort of helps the activity of these enzymes, okay? So this is the role of magnesium. So again, a magnesium, it plays a role as a cofactor, okay, to have this kinase, hexokinase and glucokinase to what? To lower the activation energy or to increase the rate of the reaction. That's what this magnesium does, okay? It is a cofactor, all right? Now, when the hexokinase puts the phosphate group into this glucose molecule, we no longer call them glucose. What do we call them? We call them, let's write down here. We call this compound what? Glucose, okay, 6-phosphate. That's what it becomes now. It becomes a glucose 6-phosphate, all right? Now after that, what's going to happen is that this glucose 6-phosphate, what are they going to do? They'll see another enzyme which is located in the cytoplasm, that is called, basically, they're going to go to isomers, right? That's what it's going to do. So basically, we call them, okay, phospho, let's say this is phosphoglycerate isomers. And isomers basically means is that you're rearranging the bond. That's what it is you're doing. So when you're rearranging from this glucose 6-phosphate, there's a phosphate group in the 6 sugar. But what happens is that, instead of, because this glucose 6 it was aldehyde sugar, they convert into, because they change the chirality. like the carbon. So what they make something called fructose 6-phosphate, make a ketose sugar. That's what they make. All right. So you make what is this compound? Which is called fructose 6-phosphate. This compound is fructose 6-phosphate. This is a glucose 6-phosphate. This is a glucose right here. Okay. Let me write on glucose with this. That's what you have to do. All right. Now, before we go like down below, let's talk about, because this hexokinase and glucokinase, this is like one directional pathway, okay? It is not reversible. This is irreversible path. Only it can go one direction, all right? So we have to talk about. Look, this hexokinase, couple of things. If I make a diagram right here, right? And we can say this is like, look, let's say this is the concentrations of glucose right here. And this is, we can say this is the velocity of the reaction, all right? Whenever we say this, a rate of reaction, whatever you want to say. You know, even in the lower concentrations of the glucose, what is going to happen? The hexokinase can go up like, and then it can rapidly, reach its maximum level. That's what it does. So basically what I'm trying to say is that, this hexokinase is, what is the importance of the hexokinase is that even with the low glucose, it can tightly bound. Okay, so that means that it has a higher affinity for glucose. So let's write down for the hexokinase. All right, hexokinase, it has a higher affinity, increased affinity to bind to the glucose. All right, which means that even at the lower concentration of glucose, it can reach its maximum. It can reach its maximum level. What does that mean? Which basically means that even the low concentration, it can bound with the glucose and it can rapidly go and it can get saturated. That's what it means. So, which basically means is that, these guys have lower Vmax, all right? That's what it means. Also, if they have a higher affinity bound to the glucose, they also have something called, low Km, all right? Which basically means is that in a very, very low glucose, all right, the 50% can get saturated very quickly, but rapidly. That's what it means, all right? And whenever I talk about your glucokinase right here, glucokinase, what happens is that with the lower concentration of glucose, what is going to happen? These guys don't really start, all right? Which means that means, they don't really function. That means what is it? That shows that they have a lower affinity for glucose, and then, as the glucose concentration increases, what happens? Their Vmax also increases, and so as their Km, all right? So here, your Vmax also increases, and also their increased level of Km, all right? These are highly regulated steps. You have to remember this. This hexokinase, all right, this can get inhibited by this glucose 6-phosphate, all right? So if the glucose 6-phosphate is like going up, like if it's going up, then what is going to happen? You don't want, because you're going to overwhelm the hexokinase systems, so the hexokinase will get inhibited because the concentration of glucose is going up. But in that case, the glucokinase will play a role because glucokinase is not inhibited by glucose 6-phosphate. Instead, this glucokinase is inhibited by this fructose 6-phosphate. So we can make this arrow here, and then we can say this inhibited right here. And then this glucose 6-phosphate can inhibit the hexokinase. All right? Because whenever the fructose 6-phosphate, this level goes up, and this can actually decrease the activity of glucokinase. All right? Now, after this, after fructose 6-phosphate, all right, we have to go back and talk about this. There's an important step right here. Let's just make it different here. Important step right here. And what is the step? And we just have to let's just say this is, we're dividing this part, okay? Now, there's a special here. What is going to happen here is that you have to convert fructose 6-phosphate to this important compound. And what is that? Is that, because we're trying to make more energy, there's an enzyme, all right? And let's just make this a very important enzyme right here. Okay, how do I make this? All right, look. And what is this enzyme? This enzyme is called, all right? This enzyme is very important. This enzyme, what it does is that it makes, okay, it's called fructose. Sorry. This is called fructose kinase number 1. This is a highly regulated enzyme, and you have to remember. Because what this does is that, it puts, phosphate group, look, already in the 6 carbon right here, there was a phosphate right here, okay? But what it does is that it puts one more phosphate in carbon number 1 here, right here. Because it's 1, 2, 3, 4, 5, 6 here. And here it puts another phosphate group here. So that's why we call them a phosphofructokinase 1. Phosphofructokinase 1, putting a phosphate group in the carbon number 1. And where is that phosphate come? Obviously come from a, look, let me use different color. You use ATP here and make, that's what you do. So basically this is a second, okay? Because this is what the first reaction here where you use ATP. This is a second reaction where you use ATP right here, okay? To put another phosphate group into the carbon number 1 position. And what do we call this compound? This compound is called right here, we call them, look, basically fructose 1,6-bisphosphate. It's called bisphosphate, right? Because the reason why we call it, it's not called bi, it's called bis. Bi means there are 2 phosphate group and then at separate positions. Because there is the 1 and other one is at the 6 position. That's why we call them fructose 1,6-bisphosphate. All right? Now, and who is doing that? This enzyme, which is a phosphofructokinase 1, PFK1 enzyme. And this is also the rate limiting steps of your glycolysis pathway, right? Now, obviously there's obviously you don't want to forget magnesium is also there, right? Now, when you're running the glycolysis, what is happening here is that, let's say 90% of, glucose probably will run into this process. It will convert into fructose 1,6-bisphosphate. But there is about 10% of glucose. What happens? I mean, this fructose 6-phosphate, what is going to happen here is that when these guys are accumulating, right? Out of 100%, let's say 90% will go and make fructose 1,6-bisphosphate. But then some of them will, you need a certain time for this enzyme to work. So what this fructose 6-phosphate do is that they'll start converting into some other enzyme. And what is that? Let's write down here in this column. That is called, let's write down here, all right? Which is basically makes another compound, all right? We'll make, look, 1 here, 2 here, 3 here, 4 here, 5 here, 6 here, right? And obviously, there's a PO4, 3 minus in this guy. But what it does? Instead of putting phosphate in the carbon number 1 position, it puts a phosphate group into carbon number 2 position, like right here, 1, 2, 3, 4, 5, and 6, right here in this. So we cannot call this fructose 1,6-bisphosphate. So what do we call them? We call them fructose. This is called fructose 2,6-bisphosphate. Isn't this crazy? This is awesome, right? And guys, this is really, really important. Because this is allosterically stimulate this enzyme, which is my phosphofructokinase 1, okay, allosterically, because it can bind to the active sites of this phosphofructokinase 1 and can stimulate this enzyme to, run the process very quickly. So it is a powerful regulator of this phosphofructokinase 1 is your fructose 2,6-bisphosphate. Allosterically, it will regulate this enzyme. And what is this enzyme which converts fructose 6-phosphate into fructose 2,6-bisphosphate, right? And that is, look, that enzyme right here is called, look, phosphofructokinase 2, right? Biochemistry is so easy when you know the information, right? So this is phosphofructokinase 2. That is the one that's going to convert your fructose 6-phosphate into fructose 2,6-bisphosphate, okay? But remember, majority of the, this fructose 6-bisphosphate will convert into fructose 1,6-bisphosphate, but some of them will convert into fructose 2,6-bisphosphate, not phosphatase, all right, bisphosphate, and that fructose 2,6-phosphate is going to allosterically regulate this enzyme, fructose phosphofructokinase 1, PFK1. Not only this, whenever there's energy level is low, let's say, I don't have any energy. That means if I don't have any, what do I need? I need more energy, right? So even in that case, right, this glycolysis run, we want the glycolysis to run. So, what is remember, there is this something called AMP, adenosine monophosphate. Look, let's write down here. There's AMP, there's ADP, and then ATP. Look, AMP, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, right? So this AMP is the lowest energy molecule, right? So if you have a low energy molecule, what are you going to have? That can also regulate this enzyme, phosphofructokinase 1. All right, but let's say your energy is going up. You just ate lots of food, right? And you have a lot of energy. If you have a lot of energy, do you want the glycolysis to run? No, right? So in that case, when you have a lot of this ATP generated, all right, that means that ATP can, okay, let me just write it down in a different color right here. Okay, let me write it on pink. ATP right here. ATP, let me write it down here. ATP can inhibit this enzyme. Inhibit. Or high level of citrate can inhibit this enzyme too. Allosterically inhibit this enzyme. So these are allosterically inhibitors or activators of this phosphofructokinase. But at the same time, you want to remember this. Insulin and glucagon, these are hormones, they also stimulate this phosphofructokinase 1. And how do they do that? So if I just quickly talk about you guys, there's going a lot of here, so I want you guys to pay attention to this. Whenever there's a high energy molecule, so when you have glucose, what is going to happen? Glucose is going to go to the beta cells of the pancreas. So when it goes to beta cells of the pancreas, right? What it's going to do is that they're going to activate, because it is a high energy molecule, that's going to induce, or that's going to cause the release of the insulin. And insulin is going to, what insulin is going to go? Insulin is going to go to the cell, right? Insulin is going to come to the cell. Let me say this is my cell right here. And then here, insulin has a receptor right here, right? These are called enzyme-linked receptors. There's a tyrosine kinase receptor. And when the insulin comes, let's write down here, insulin comes here. It binds to these enzyme-linked receptors, right? And that actually the tyrosine kinase receptors, it binds to the insulin, and with a bunch of steps, right? What is going to happen is that they're going to activate this enzyme called like a phosphatase enzyme, right? And what they do is that, they'll come and remove a phosphate group. This is really important. The phosphate group. And whenever, from this phosphofructokinase 1, whenever you remove phosphate group from the phosphofructokinase 1, meaning whenever you do dephosphorylations, then this enzyme, which is a phosphofructokinase 1, that get activated. It is a very important concept here. Again, I'll repeat. Whenever the insulin will activate this enzyme, which is a phosphatase enzyme. When that phosphatase comes and then will remove the phosphate group from the phosphofructokinase 1, that will cause this enzyme to be activated. And now you have to know who put the phosphate group on the phosphofructokinase 1. That is glucagon. Because glucagon will come, and with the GS pathway, it's going to activate the adenyl cyclase and activate protein kinase A. And that protein kinase will put a phosphate group into the phosphofructokinase 1. When the phosphate group is attached to this, okay, when attached to this enzyme becomes what? This enzyme becomes inactivated. That's what it means. Okay, so by putting a phosphate group, all right, this enzyme becomes what? By putting a phosphate group into your enzyme, these enzymes become inactivated, right? But by dephosphorylating with the insulin, what is going to happen? This enzyme will become activated so that you can run the glycolysis. And that's the hormonal way to regulate this PFK1 enzyme. All right. Now, after this, what do you make? You make a fructose 1,6 bisphosphate, right? That's what you make. And from the fructose 1,6 bisphosphate, this is where these 6 carbon molecules that get split up into 3 carbon molecules. So let's meet. What is the enzyme called? It's called Aldolase enzyme. It basically breaks down into 3 carbon molecules. So if you follow me, this one, this is 3 molecules carbons, and you have one carbon right here. This is a carbon number 1, 2, 3 here. This is where you're going to find a phosphate group right here. And then if you see, look at this right here. This is again, if you split in 3, would have 1, 2, 3, and another would be in the third position. You're going to find a phosphate group here. We just like break down into this. That's what we did with this aldolase enzyme. So when you split up these 3 carbon molecules, based on the phosphate group attached to it, we give a name. Okay, and these guys right here, we call them a. Let me write down here. We call them a dihydroxy acetone phosphate, or simply you can call them a phosphate, right? Or simply you can call them a DHAP, dihydroxy acetone phosphate. And what about this compound, which is basically called glyceraldehyde, okay, glycerol, aldehyde, 3-phosphate. The 3-phosphate because in the carbon number 3, there's a phosphate group. Or you can simply call it GAP. That's what it's called. So DHA, DAP, DHAP, and then GAP3 that is formed by this enzyme called aldolase enzyme. All right?